All right, we is back. And we talked about how do neurons send signals. The main way they're kind of thought of is electrical, but then hopefully I convinced you guys in some way that they also send them via chemicals, such as sodium, potassium. And it's also a mechanical process, such as the sodium potassium pumps, as well as the snares, fusing the vesicles, things bumping into things, channels opening, blobbity bloobity blop. Okay, let's now talk more about sending these signals. So let's just draw, you might want to redraw just this neuron here. And we're going to talk about a couple of ways that signals are propagated or messed up, kind of how signals are facilitated in being sent. So all along this neuron, there's actually other cutesy little cells like these, all along it. Very cute indeed. And these guys are like the little tortilla that I was talking about. So they'll look like that all along the axon. And they'll have their little nucleus there too. Bloop, bloop. And there's actually another cell that kind of comes down and forms all these around it. Don't worry about that right now. Oligodendrocyte. But these guys here are called Schwann cells. I like that name. Schwann cells. Reminds me of the Schwann's truck that would come and deliver weird frozen food to my grandparents. Schwann cells. It's kind of like swans. And what do swans got? A big old long neck. All right, well, if we take these cells and we actually look at them like this, so we do say a transverse cut through them. So this is the axon, and we cut it, and then we look at it like this. We look at it end on like that. And then we're going to look and see what's going on. So the center here would be the axon. And then the Schwann cell, it's actually wrapped around it. A little bit something like this. I'll do it in a regular pen, I think, maybe big. So it actually wraps around it a bunch. Like so. Like a tortilla. That's why I was eating that tortilla, not because I was hungry. Okay? Like that. So the Schwann cell wraps around the axon multiple times. And the function of the Schwann cells is to insulate the neuron. Just like plastic around a wire. You could think of Schwann cells. Another name for this is the myelin sheath. Myelin sheath. So I'm sure some of you have had like a cord, a phone cord or electrical cord or something get broken or frayed, or the plastic comes off or something. You see the wires underneath it. Well, why do we insulate a wire? Well, because there's electron movement going on in there. There's a charge going on, and we want to keep it in there. We want to insulate it. Okay, that's what these do as well. So they allow to insulate the neuron for optimal electrical activity. What's up, birdie? It's a little birdie right here. Let's see if you can see. Oh no. He flew away. Bummer. Okay. Okay, so Schwann cells. All right. They insulate the neurons. So for example, we can't have leakage of the ions. And what this actually allows to happen is instead of having to have these ion channels all along the length of the neuron and having to wait for all of them to open and close and blah, 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 blah. That would take forever. What we can actually do is the signal actually jumps. I mean, it would be doing it inside. If I'm just drawing it outside, it actually jumps along the neuron. Because you only have to have sodium and potassium jump or opening and closing right here where it's exposed. And then it can kind of skip through this because the membrane potential will propagate along the membrane, open up this channel. And then that channel opens and the membrane potential will propagate along and open up this channel. So this actually speeds up transmission as well. So the myelin sheath also speeds up nerve impulse transmission. How fast it goes. It speeds things up. For example, mollusks like squid and octopus, they don't have myelinated neurons. They don't have Schwann cells around their neurons. Okay. All right. It also helps protect your neurons and things like that, but it helps prevent like ion leakage, so it insulates them, and then it helps speed up. It also reduces like the cost, so it's more efficient. More efficient. Less channels are needed, right? We got less ions to pump. If we only got to pump ions here, 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 it's a lot less pumps, it's a lot less channels, it's a lot less ions. Less channels, ions, pumps needed. Pretty nifty. Thank you, Schwann cells. Okay, well, some of us are real unlucky. You guys remember white blood cells, right? Well, sometimes our white blood cells are part of our immune system, and sometimes they don't function properly. So we got a white blood cell here. Kind of looks like a ghost. It's got a nucleus. Let's fill that nucleus in. And sometimes we have things called autoimmune diseases. So this is a white blood cell. And what it's going to do is it's going to incorrectly think that this Schwann cell is an enemy, and it's going to attack it, maybe with some enzymes, digest it, and eat it. So white blood cell attacks Schwann cells, killing them. Thinks it's some kind of invader. What you doing on my neuron, boy? I'm gonna kill you. That's no good. All right, this is the cause of a terrible disease that you don't want. Multiple sclerosis, which is basically like the degradation of your ability to control your own muscles. And a member of my family has this. You don't want it, I promise you. And that's caused by our immune system attacking our own body. And it's specifically attacking the Schwann cells of our neurons. Okay, so that's one disease I want to talk about another disease here also that has to do with nerve impulse as well. You may have heard of this one. All right. All right. So this next one, first, let's just talk about a little anatomy here. I'm going to delete this guy actually. Get rid of him. We're going to talk about Alzheimer's. I'm sure you've heard about this one. Alzheimer's disease. All right. This one's very interesting. So actually, if we go back, it's funny how when we talk about the central nervous system or the nervous system, I just want to talk about cells. You see that? See how I trick you? Well, look at this. We're going to talk about the cytoskeleton again. Oh boy. We got microtubules. Remember these? These are what little motor proteins walk on. They help deliver things throughout the cell, like vesicles and stuff like that. Also organelles. And then we've got actin. This one's we find in muscles, but it's also important for kind of changing the shape of our cells. All right. Well, if you look at neurons, they are very beautiful on the inside. Beautiful little souls. Whee! Okay. Well, if you look at a neuron, they look something like this. In its axon, they got microtubules that go along the center like that, and then they got rings of actin. I'm not going to actually make it a ring. That go all around it and decorate it kind of like this. So these would be going around kind of the length of the axon. So we've got actin rings. Actin rings. And kind of help shape the neuron. And then we've got microtubules. Microtubules. That allow, this thing is freaking long, dude. Like, look how long that thing is. It could be up to 1 meter long. And the cell's only got one nucleus. So you got to be able to ship things all the way down to the end of it. It's pretty crazy. Because you certainly still need RNA and mitochondria and all that stuff down here. Okay? Sensory neurons kind of go around this and they put their cell body right in the middle. But motor neurons don't do that. All right. Okay, so you got a boatload of microtubules and actin rings in here. All right, well, microtubules are actually also decorated with this little guy. There's other proteins with them. There's motor proteins, all that jazz. But then there's also little things like this. They're very pretty. They kind of come off of it. They look like little kind of like bent staples. Like this. And it's called a tau protein. Tau protein. Okay, and these would be like all over the length of the microtubule. Like this. They look like little bent staples coming up off the microtubule. Okay? All right, well, let's go back to this guy. Oh, look at this. This is in... Oh, yeah, look at that. We'll get to that. That's leftovers. Okay, so let's redraw this guy. Stop me when you know it. It's always green. Looks like a stack of pancakes. Like this. And one of the labs at the University of Michigan that I was right next to, they actually studied this process that I'm telling you about, which is why I know anything about it. It's because I was next to a lab that studied this. Okay, this, if you remember, is our Golgi. Little vesicles butt off, like so. And they transport proteins, right? or lipids up these stacks, and they help fold them up and modify them, add sugars to them, do whatever they need to do. All right, well, let's say we start causing shipping and handling and modification problems in the Golgi. All right, that's crazy. So we start messing this all up. And now proteins that are going to be going out here, getting shipped around, doing all kinds of stuff, get messed up. We start messing up proteins because of this. All right. And one of those proteins we can mess up is this guy called the tau protein. Okay. And one of the things that will happen when we mess it up is something like this. I'm going to draw it over here. So instead of binding to microtubules like that and doing its little thing, what it would rather do, I'm going to draw this little size of the microtubule. So it would rather now, well, I'll actually draw in the kind of mutation kind of thing. Maybe now you have two taus looking like this. Okay. So now two tau proteins strongly bind to one another. Okay. If you remember all the way back here, these are actually beta sheets. Oh yeah, I'm gonna do it, guys. Let's go all the way back. See how all this is connected. We're gonna go to organic chemistry here. Remember these? Oh, we got a peptide, protein peptide, folds on itself, and we got two secondary structures, beta sheets, back and forth folding, or we have alpha helices, which form little spirals. So this back and forth folding beta sheets, if you have those from two different proteins and you make it really strong, it'll be tough to break those apart. Beta sheets, very strongly associated with one another due to hydrogen bonding. Okay? Lots of little hydrogen bonds would now hold these tau proteins together. Okay, you want to know how strong it is? This is called an amyloid plaque. All right. Another amyloid is spider silk. Spider silk is made out of a protein and it is an amyloid. It's beta sheets connected together like this in a peptide. Very, very strong stuff. They make bulletproof vests out of spider silk or whatever. This tiny little thread that you can barely see can catch bugs and prevent flies. It's super strong. Okay. All right, so now you get a bunch of these tau proteins together. Boom. I'm going to make them a little smaller. I'm going to do a little copying. I'm going to do a little rotation too. See how that looks. Oh yeah, this is coming together. Oh, perfect. Yeah, that's good. I could just keep copying that. Boom. So now we just keep making tau protein because tau is not doing what it's supposed to be doing, so the body thinks it needs more. And it just keeps sticking to itself. It's mutated. It's not doing its job where it should be on the microtubules. And instead what it's doing is accumulating all up in the cell. Oh boy. This is bad. Oh, this is really bad. Okay. My computer doesn't even like this. You see how slow it's kind of getting? It's sluggish from copying all of these tau proteins. These are now called amyloid. These would be the amyloid plaques or the tau tangles. Oh yeah, look at that. It's just kind of insane to even look at this. Amyloid plaque, or it's also called a tau tangle, or a tau tangle. Okay? And that's what causes Alzheimer's disease, is this accumulation of these tau proteins. And this would accumulate not just here, but like also here. It'd be accumulating everywhere. Okay? And what needs to kind of happen when a channel opens up here? Sodium ions need to diffuse and get through this now. How could they do that? I mean, look at this. This is a nightmare. This is what nightmares are made out of. Sodium ions are going to get through this? Okay, maybe, but it's going to take longer. It's going to slow transmission in your neurons. And maybe you get enough of this, and the neuron can't even function anymore. And that's what causes Alzheimer's. So Alzheimer's is caused by misfolded proteins accumulating in what are called aggregates. Special word for that is amyloids, which inhibit neuron signals from being sent properly, and can result in neuron death. And it's a progressive illness. It gets worse as it goes on, and you see why. So now we got problems sending signals. Maybe now we got even more Golgi problems. We got more protein folding problems. We got more aggregates. All right. It's bad. It's real bad. Okay. That's Alzheimer's. Okay, let's keep talking about signaling. That's kind of, got kind of dark. Let's keep talking about signaling. How are we doing on time? I should probably just keep going. Let's just keep cruising. These will all just be a little long. Deal with it. Okay. All right, let's move down here. Let's have a little fun because we had that was a little scary. We talked about multiple sclerosis and Alzheimer's. No, thank you. Okay, let's talk about something fun. This is one of my favorite things about the nervous system. Okay, so here we're going to look at electrical activity of a neuron. So we can take electrodes and stick them to a neuron. So we'll draw our little neuron here. He's so cute. Okay. So we got a little neuron. Oh my God, he's cute. Okay, we got 3 of them. Okay. We can take a little electrode. What color? We'll make it blue. We can take a little electrode. And we can attach it right here. And we can see when the neuron fires. So this will be an electrode, just a wire. And we can stick it right onto the neuron and see when it fires. We, of course, have one of these for each of them. And we can look at a normal neuron, and this is actually a special neuron called a mirror neuron. We can measure the electrical activity. So here we'll have electrical activity. That'll be kind of going up here. Electrical activity. All right, so then we can measure the electrical activity of these neurons. And we got 3 different scenarios here that I'm going to talk about. Let me move these down a little bit. Okay. So what are these scenarios in which we're measuring electrical activity for these neurons? Let me pull these guys down, because I got to do some fun drawing. Okay, we are going to draw some fun stuff here. So we got to see how I do here. Yeah. Oh, baby. Uh-huh. Oh, yeah. Oh, yeah. Pretty good. Pretty good. Oh, yeah. All right, that ain't bad. That's my monkey. Clearly, that's a monkey. And we put him in a box, so he can't move, because monkeys be crazy. And then we have his little arm sticking out, his little monkey hand. I gotta redo that, but that's fucking perfection. Okay. Dude, I cannot. All right, that's his monkey hand, obviously. Monkey hand. All right, and we're looking at a mirror neuron. And mirror neurons are associated with learning and empathy. Very interesting stuff here. Learning and empathy. I don't know why those eyes made it so much better. Okay, so we got this monkey hand, and the monkey is at rest. Monkey is at rest. The monkey just chilling, bro. All right, and we look at this electrical activity of this neuron, and it's just flat. It ain't firing. Neuron is off. Okay. Now, we take our same monkey with our same perfectly developed hand, and we have that monkey squeezy a ball. Squeezy a ball, pleasey. The monkey squeeze a ball. Monkey squeeze a ball. Ooh. Monkey like a squeezy. All right. Monkey likes to squeeze the ball. All right. That part's not actually important, but I bet it does. Okay, then the neurons, every time it squeezes, the neurons are going to fire. So it's going to be like whoop, whoop, whoop. Okay. Every squeeze causes the neuron to fire. Pretty cool, right? We can measure the electrical activity every time it squeezes. All right. Now, what we do is we take the monkey. Monkey no squeezy ball now. Monkey still have hand. Monkey may be a little mad. Okay, because monkey's just sitting here. Obviously. That's a human hand. Monkey is watching human squeeze ball. Okay. Monkey watches human squeeze ball. What do you think happens? Does this neuron fire? The answer is yes. So just by watching the the human squeeze a ball for an activity that it's done causes the monkey's neurons, mirror neurons, to fire. Every squeeze of the ball by the human causes the monkey's mirror neuron to fire. Pretty interesting. This is like why it helps to like do something, watch somebody else do it, you do it again, then you watch somebody else do it, then you do it again, then you watch somebody else do it, because you're learning from both of those processes. So mirror neurons, very fascinating. Mirror neurons help us to learn and cause empathetic responses. I'm a pretty empathetic person. Somebody starts talking about, I don't know, one of their family members dying or being diagnosed with cancer or overcoming something, or I relate heavily to people being accepted or accepting others for who they are, or overcoming terrible odds and abuse and situations and things like that. That will heavily trigger my empathy, and sometimes it will cause me to cry. Even though it's not happening to me, and it's not happening to them currently. And that's because my mirror neurons are firing. We talked about our rats being in sync. It's pretty cool that we have neurons like this that will fire in synchrony to this kind of stimulation, even though we're not the same person and we're not even touching each other. We're just watching or being in the same room or listening. Pretty nifty stuff. That's a little bit about mirror neurons. Nobody else in this college is teaching you about mirror neurons, so there's a little bonus for you. Okay, now let's talk about all right. I'm trying to think how I want to do this because I got a funny thing here. Do I want to end with the funny thing, or do I want to? Let's go with the boring thing. Oh God, I really sold this part. It's the boring thing. All right, what we're going to do is look at the spinal cord transverse section. So we're going to cut it in half, and then we're going to look at it end on. So we're looking at a spinal cord here. And you'll see this in lab when we get to this section. So we got these ganglions that kind of stick off the back of it. And this is actually pretty fascinating. I don't mean to undersell this. I don't think this is boring. It's just not. It's not as entertaining. Okay. These are ganglia, these little things sticking off the spinal cord. This is the spinal cord we're looking down at. We cut somebody in half and we're looking down their spinal cord. Okay, and it kind of looks like a little like butterfly here in the center. Something like this. Not too shabby. So this is here gray matter, and this is actually white matter. So this is gray matter, gray matter. Make that real small. You can label it out there if you want. A birdie bag. And this is white matter. All right. Gray matter and white matter. And your spinal cord, like I said, is it does a lot of thinking for you, I guess is the best way to put it. All right. All right. So now let's draw my beautiful supple leg here. Obviously, I got some trifling feet. That's my ankle. That's my beautiful calf. Oh, baby. So good. Okay? In Uchis, I step in attack. Tori steps on attack. Dun, dun, dun. This is the story of that. What happens? Tori steps on attack. Well, if you remember, got two sets of neurons that are coming in and out of the central nervous system. We got sensory neurons and we got motor neurons. So we're going to have our sensory neuron down here, which is going to have, dendrites coming down like this. And it's going to sense that, and it's going to send a signal all the way up. It's going to continue up from my little tootsie up through this dorsal ganglia. Cell body is actually right there. And then it's going to have an axon right there. So this would be the cell body, sensory neuron, of the sensory neuron. Let me just give a little thing. So this would be the dorsal side, dorsal, or wait, posterior. Sorry, we're talking about this. We call this the dorsal root ganglia, but then it's the posterior side of a human. Posterior. This is posterior. What's the opposite of posterior? Anterior. So your sensory neurons come in the back of your spine. All right, so this is a sensory neuron. Sensory neuron. Sends signal to spine. All right. Okay. Okay, and then we have a little guy in the middle here called an interneuron. So the interneuron would look something like this. So it would have its dendrites here. And then it would have a cell body. And then it would have an axon. Like this. Okay, so this is a cell body of a sensory neuron right there. Cell body. This is an interneuron. Interneuron, because it's in between things. An interneuron. Okay. And then we've got, coming off of this bad boy, what are we going to have? We're going to have the dendrites of another neuron, and this is going to actually be a motor neuron. And remember I said some neurons have things that are like a meter long, right? This would potentially be one of them. Going to my leg. So this axon's full of microtubules. And maybe this goes to like my calf and my quads and all kinds of stuff. And then we have a neuromuscular junction right here. It releases an action potential, which then releases calcium from the sarcoplasmic reticulum, which then moves troponin and tropomyosin. So then myosin combined to actin, and then you have ATP and power strokes happening, and you get your leg to move. So this is a motor neuron. Motor neuron. It's connected to your skeletal system. All right, motor neuron. And here you see that we actually have, in the gray matter, we have the cell bodies. So the cell bodies are in the gray matter. Gray matter contains cell bodies. And the white matter contains axons. And that's why it looks the way it does. White matter contains axons. Gray matter contains cell bodies. This whole process, so then signal to muscles to move leg. Send signal to spine about ouchie. Ow! Sensory neuron sends signals to spine about ouchie. Motor neuron signals to muscles to move leg. This whole little jam thing cycle that we drew here is called a reflex. And the next time you're walking around, I want you to appreciate the fact that you aren't thinking about walking. Your body is walking for you, and that's because it's a reflex. You have sensory neurons that measure the stretch. They sense the stretch of all different parts of your leg, tendons, muscles, everything. And they say, okay, now it's time to move a motor neuron because we're walking in this direction and our leg is in this now stance, it's being stretched. Okay, we need to contract it. The only thing you mentally, consciously control about walking is when you initiate it and when you change it. Like, okay, now I want to walk faster. You send a signal kind of to this system that says, walk faster, and then the reflex takes over. And we'll test this stuff in lab. I'll get to hit you guys with a hammer. It's my favorite part of the class. Okay, let's end with a banger. And this ties into something that we've already learned. All right, let's draw a very simplified central nervous system here. Okay, so we got a little brain here, and we got a spinal cord. Okay, so we got a brain, and we got our spinal cord. And we've got two types of nervous systems. Okay, we've got a sympathetic, and we've got a, I like this blue, it's kind of, what makes this actually red? Sympathetic, and then we've got parasympathetic. Okay. All right. Well, let's talk about this. Let's talk about this word right here. What is a simp? A simp is someone who is pathetic, right? A simp is someone crushing super hard for somebody. An SIMP, right? You don't want to be a simp. You don't want to be simping for somebody that's kind of pathetic. And when you're simping hard, you're way too excited about somebody. You're way too excited about someone. Just like, come on, bro. Quit simping so hard. She ain't never gonna be with you, bro. Take the cape off now. All right? And it's pretty pathetic to simp that hard. Are you with me? Way too excited. What happens when you're excited? Heart rate goes up. Breathing goes up. Your sympathetic system is your fight or flight system. Fight or flight. This is, all right, business getting real. Business getting real. All right, so this causes our heart rate to increase, heart rate to increase, heart rate to increase, respiratory, breathing rate to increase, constricts our capillaries, or the ends of our vascular system, to keep everything in the core and on the big muscles where it needs to be. Constricts our capillaries and inhibits digestion. So that's why, like, if you're really stressed all the time, you're constantly in fight or flight, and you're not properly digesting your food. It's pretty crazy. And these neurons that come out of the spinal cord, they all kind of come out in the middle. It's pretty convenient for us, like anatomically. So the neurons that control these all kind of come out in the middle. I'm not going to draw all of them. If you get into anatomy, you'll need to know more of these. All right, now we got parasympathetic. And parasympathetic, okay, you're still a simp. This word here, you're not a simp. This one is you're around. That's what para means. It means around. You're around your simp. Who is pathetic? Right? They're pathetic. But you're around your simp. Dude, if I'm around somebody who's like, crushing on me and in love with me, yeah, it feels pretty good. I feel pretty calm. It's like, all right, well, I mean, if like a lion comes up, I'd be like, hey, simp, go get eaten. Hey, simp, go fix me up some mostaccioli and meatballs. I don't know if people know what mostaccioli is around here. It's a Michigan thing. Give me a punchki. Give me a coney dog. Anyway. All right, so what's going to happen here? Our parasympathetic, you're around your simp. You're very calm. You are calm. This is your rest and digest nervous system. I'm just going to write system. Rest and digest. Fight or flight, rest and digest. Sympathetic, sympathetic, heart rate increasing, breathing increasing. Rest and digest. My simps are around me. Go give me a punchki. All right, so it's basically the opposite of this, right? Heart rate will decrease. I'm going to move this up here and move this one up here just so it's out of the way a little bit. Okay. Heart rate will decrease. Breathing will decrease. It will dilate your capillaries, dilate your capillaries or capillaries, dilate capillaries, and stimulate digestion. And stimulate digestion. And those will actually all come out of your neck. And it will also do things like, there's actually a couple that come out of here too that like control your rectum and bladder. Like if you're super amped up, it's like hard to pee, right? You gotta like if you have anxiety, if people have anxiety about like going to the bathroom in public places, they sometimes they can't because the anxiety is so strong. It's like your sympathetic nervous system is saying, dude, get your simp around you. If you want to pee in a bathroom, get your simp around you. All right. These all kind of come out of kind of the cervical region up here, which is pretty crazy that your digestive ones are coming out of there. Let's look at these. Yeah, so these are kind of the neurons that we're talking about here. All of these going in here. Anyway, I'm not actually sure about that. I'm not actually sure where the digestion ones are coming out of. Don't worry about it. We'll put it right there. I'm actually not sure on that. I don't want to say anything wrong. But it does stimulate digestion. I'm just not sure where they come out of. Okay? Just so you're aware. Okay, so simps are pathetic. They have a high heart rate, high breathing rate because they're crushing on you hard. I know you all got at least 5 to 6 simps in here. And when you get them around you, you're like, all right, go give me a punchki because I want to eat. If you don't know what a punchki is, it's this. Paxi. Let's see. Yeah. It's a Polish thing. These are hot in Detroit. They are delicious, so I recommend getting some of these. There's a day in February where everybody eats like 6 of them. Yeah, Polish Paxi, but you say it punchki. It's a punchki, brah. It's a punchki! Get your punchki on. Mmm! Look at that. They're full of jelly. Brr. Ugh. Yeah. I want one now. Let me know if you've eaten a Punchki. Okay. I'll see you in the next one. We got probably 2 more. Sorry. I like this stuff. Later.